Now it's going.
Okay. Hi, Mr. Masters. This is my Chapter 13 project on protein synthesis, which is the process of transforming DNA into RNA and then into proteins. So, how the definition of transcription is using segments of DNA to serve as a template to produce complementary RNA. So, I drew a diagram of the transcription process, and as you can see, this process takes place in the nucleus, and it shows how the RNA polymers, which is an enzyme that will bind the two together and make the template, separates the DNA strands and puts together the RNA strand and the DNA strand so that the RNA can form into, mold into a template for later use in translation. Then over here, I drew a close up of what is over here. So it shows the bases on the DNA strand and the bases on the RNA strand. So it shows how A is the opposite of U, T is paired with A, and then C is paired with G. And as you can see that there is no T on this side because T is not in RNA. After it goes through this process and the complementary RNA strand is made, it goes through RNA editing, which is taking out the unwanted parts, which are called introns, and leaving in the wanted parts, which are exons. So how this works is it starts as DNA, and then as it's going through this process, it goes into, which is called pre-mRNA. It goes, so the introns are removed, and caps are added on. And then after this process, it turns into mRNA, which will be used in translation. And it is messenger RNA, which is what carries the instructions for the polypeptide synthesis in translation. And that's it for this board. Okay, so this is the second of 3 boards, and this board talks about translation, which is the decoding of mRNA into proteins. So in translation, there are 3 different types of RNA. There's mRNA, which carries the instructions for polypeptide synthesis. The second kind is tRNA, which carries the amino acids to the ribosome and matches them with the coded mRNA message. The third is rRNA, which forms an important part of both subunits of the ribosome. And how the mRNA is read is with genetic code, which is read by 3 letters at a time, so that each word is 3 bases long and can correspond to a single amino acid. So there are 2 different types of ways you can read genetic code. So there's a codon, which is the 3-letter word for the mRNA bases, and there's an anticodon, which is the 3-letter word that is attached to the tRNA. So over here is step 1, which is transfer RNA. This diagram shows how there is the ribosome RNA, which is this whole entire squiggly box, and then it shows how the anticodon and the start codon bind together. So it begins at codon AUG, which is methane. The tRNA has the anticodon attached to it, and the codon and the anticodon match up. The rRNA positions and binds them together. And then this is the amino acid phenolphthalein, and then this is the transfer RNA, and this shows the anticodon and the codon binding together. So the second step is called polypeptide assembly line, which shows the RNA binding the 2 amino acids together with a peptide bond to form a polypeptide chain. The tRNA and the anticodon break apart, shown right here, and the tRNA floats into the cytoplasm. The next tRNA floats in and will attach to its codon, so shown here. And then the final step is completing the polypeptide. So in translation, the polypeptide chain stops when it reaches the stop codon, UGA, which is called stop, right there. And then the polypeptide chain is complete and the proteins were produced. So this is the polypeptide chain, and it is shown that there are peptide bonds in between, and this shows the last tRNA that completes the protein. Okay, that's it. Okay, so this is my third board. It's about mutations. And mutations are heritable changes in the genetic information, and there's two kinds, gene and chromosome. So this is what an average gene looks like with its genetic information. That is what it looks like regularly. And the first type of mutation is substitution, which is the changing of one base into another. And I marked the mutations in purple marker. So what was replaced was, instead of a C base, it was a T base in the genetics, which results in a different amino acid, but it doesn't affect the overall protein as much as some other things. It just might affect the protein function a little bit. So the second type is insertion, which is when an extra base is added to the DNA sequence. So what I showed here was that the extra base A and U were added to the DNA sequence, which resulted in this over here to be thrown off. So the pairings are all different, and as you can see, it resulted in 3 different amino acids instead of just one up here. So it has a more drastic change. And the third kind is deletion, which is when a base is removed from the DNA sequence. So this squiggle line represents the base G with its paired with C was removed, so it thrown off the genetic sequence. So there is no base for this part, which throws off the amino acids, and this one you don't even have an amino acid. And for over here, the chromosome mutations, this is what it looks like before mutation, as I've illustrated here. And the first type of mutation for chromosomes is deletion, similar to what it is over here. But in this, it is a loss of all or part of a chromosome. So chromosome B was removed from the sequence. The next type is duplication, which is an extra copy of all or part of a chromosome. So I illustrated that there are 2 B chromosomes instead of just 1. The third kind is inversion, which reverses the order of a chromosome. So I showed that chromosome C and chromosome B were switched, so that can result in a different trait. And the final kind is translocation, which is when one chromosome breaks off and attaches to another chromosome. So the chromosomes G and H were added to this sequence and were added in the middle of the chromosome, which can result in very bad genetic disorders and chromosome disorders. So we also had to talk about operons. So I showed the process of a lactose operon. So right here, it shows the lactose repressor gene, which is represented in red, and this is the repressor protein. This is the operator, and this is the promoter, and these are the lactose genes. in the middle, it shows what happens when lactose is not present. So here's the RNA polymers, which is attached to the promoter, and here is the the repressor on top of the operator. So the repressor binds to the operator, blocking the RNA polymers from going through transcription. And over here, I showed what it would look like if there was lactose. So right here, it shows the polymers not being blocked by the repressor, so it is able to go into transcription, and that's the RMA. And then down here, it shows the lactose binding to the repressor, and that allows us to go into transcription. So yeah.